to warm up this weekend, and I'm looking forward to it.
I bet you are. I bet you are. Say, we want to just welcome everybody. This actually could be the biggest group that we've had pre-registration. So I think there's a lot of things that your science is examining that many of us are interested in. But just a couple business points first, guys. We are doing a new series here. We really wanted to introduce our newer researchers to our CRF family. Dr. Hume would be certainly one of those. We are recording this. This is something that is going to the cloud, so please feel free to look at it again. Share it with your family and your friends if that's something that you'd like to do. As I said, we were doing a new series, and this is a specific program that is being funded by the Boren Family Research Grants. We want to make sure we have a shout out to Danny and Cheryl Boren and their family for their contribution to make this this available. And also needed to make sure we thanked our sponsors, which would be Santen, Biogen, Genentech, and Spark. Their funding too is helping us come to you today. So pretty cool and pretty interesting. Dr. Hume and I were talking yesterday, and she was sharing some really neat things that I can't wait that all of you can learn more about the work that she's doing. It's quite profound and exciting. I'm going to take your questions maybe at the time, and if there's appropriate pauses, I will ask them. Otherwise, we'll make sure we get to everybody's questions at the end. I think that's it. I think I covered all the basics. So we're super glad to have you here. I'll let you go ahead and share your screen, and I will stop mine. Thank you, Stacey.
Absolutely. Hopefully everybody can see this. And I don't mind being interrupted along the way because we're going to talk about a lot of things today. So I have made this a science talk for non-scientists. So my intention is not to make anyone feel less intelligent, because I think some of you know a lot more about Croiteremia than I do. And so if I've made this too easy, it is absolutely my apologies. So just a bit about me and how I came to work on Croiteremia. I have run a diagnostic lab here in Edmonton for over 17 years. And probably about 10 years ago, Dr. Ian McDonald approached the lab and said, we don't have local testing for our Croiteremia families, in fact, even in Canada. Would your diagnostic lab please set up genetic testing for Croiteremia? So we did. We set up testing for Croiteremia, and that started off a collaboration between Dr. McDonald and I and his lab. So that's how the story of this begins. I also do research, so I'm not just running a clinical lab, but I also have research that I do. So this really does combine both the diagnostic lab as well as my research. Okay, so let's get started. So the title of my talk is Finding a Cure for Croiteremia Through Discordant Brothers. And like I said, I work at the University of Alberta, and I had to start my slides with the Croiteremia Research Foundation because both the Canadian and the US foundations have supported this work, and I'm forever grateful for that. So I borrowed these slides from Dr. McDonald, who has probably presented to you all before. So I'm going to get through this pretty quick because I know a lot of you know a lot more about this than I do. So we know that Croiteremia is a slowly progressive inherited retinal dystrophy, and it is quite rare with a prevalence of about 1 in 50,000 in the general population. It is a monogenic disorder. That is, it is caused by a single gene, and that is the CHM gene. The CHM gene is found on the X chromosome. Wait, it's moving by itself. On the X chromosome, and therefore it mainly affects males. Females have 2 X chromosomes. It does account for about 4% of all blindness. The classic course of Croiteremia is that one starts with night blindness in early childhood. And we can see this by looking at the images of the fundus early in childhood. And we can see that as we progress through the years, that our fundus grows to lose more pigment as we go. This part, your macula, is responsible for your central peripheral vision. And so as we lose your peripheral vision, we see that we've lost some of the pigment that is important for your photoreceptor cells to transmit light. And you can see how abnormal this gets until somebody in their late adulthood has completely lost vision. So I'm going to show you what I know you've all seen before, images of the normal fundus versus fundus that you see in CHM patients. And this is because I need to show you the fundus of the two brothers that we have identified. So we identify that you have retinal pigment epithelial changes, and you can characterize these on the images by seeing what is termed a salt and pepper appearance. And so I think Ian's slides have an automatic progression. So we can see that we're losing pigment around our macula, which means that our photoreceptor cells are not able to transmit light. So the pigment becomes more transparent, as we're starting to see, and we're starting to be able to see the deep vessels present in your choroid. So as we progress through the disease, you can see that we've lost even more pigment in the periphery, and that we have now just retained vision in the central part, as can be seen by the pigment that is still present surrounding your fovea or your macula. In a late stage male, again, we see that we have the complete absence of pigment. Female carriers, because they have two X chromosomes, usually experience a milder form of Croiteremia. Usually they get a decline of visual function characterized by night vision loss, usually after the age of 50. In some rare cases, female carriers actually can exhibit fairly severe symptoms, more like that of an affected male. And we are not sure why. Sorry, I think Ian's slides have a little, hopefully when we get into hit my slides, it doesn't do this. So we have identified two amazing siblings. We have two brothers with the same causative CHM mutation, which we now call a variant. We try not to use the word mutation anymore. These brothers are currently in their 30s and 40s. One brother has presented with classic CHM by his image on the left, while amazingly, the other brother is unaffected. And you can see here that he still has a lot of pigment. So we realized that this was a completely discordant phenotype. Both brothers have a mutation that should cause classic CHM, but one brother does not yet have it. So we realized that this is likely the only case ever identified of Croiteremia that is completely discordant at this late age. So could the cure for CHM lie in the unaffected brother? What is saving his vision thus far? How come he's unaffected? So to examine this, we're going to get into a bit of the basic central dogma of biology. So we start off with a central dogma of knowing that every cell in the human body contains DNA. When the cell decides that it's time to use this DNA, it undergoes a process called transcription to make RNA. RNA is very transient and only exists temporarily to make the final protein that is required in the cell. And the proteins can be thought of as the machinery that do the functions in the body. So in my little diagram down below, I have a picture of some machines that would be the equivalent of what proteins do. I've got RNA labels as the manual, the how-to instructions, how to make proteins. And I have DNA listed as being present in every cell in the body. The reason why RNA is transient is because you'll see here that it is single-stranded. DNA here is double-stranded. And so DNA is very stable. We can examine DNA that's been in blood off of a sidewalk for a crime scene, but we can't look at RNA because the minute RNA is outside of the body, RNA quickly degrades. I just have to put a plug in because this is a science talk, that the two COVID vaccines that are being widely distributed, Pfizer and Moderna, are based on RNA. They are not stable for long. Once they get in the human body, they make the spike protein for the COVID, and then they are quickly degraded by the body. RNA does not go back to DNA. It does not, this arrow only goes one direction. This RNA is not integrated back into the DNA. In fact, these vaccines are so safe because the RNA is degraded. So I just have to throw that we use our knowledge of biology every day, not just in Croiteremia, but actually in vaccines. So let's move on from this central dogma of biology.
Hey, Dr. Hume, can I just ask you a real quick question? We had a Brent asked if you could just answer this quickly. How would you define how old is a really early stage male and a late stage male? How do you define those two stages?
Okay, so I am not the clinical expert, Brent, in any of this at all. And so if I know Dr. McDonald is not on the line, but my understanding is that typically people will present in their teens. And even if people don't have, aren't seeing the differences in their vision, that when we take detailed images, that the ophthalmologist can see the changes in their vision. And so for us, the fact that this male brother that we have is, I think, in his young 40s right now and doesn't show any pigmentary changes is quite late. Now, I have not said that this brother would never be affected, but he's certainly much delayed over where we would have expected him. And Dr. McDonald has told me he has never seen someone with a CHM mutation that is not shown signs by the time he's in his 40s. That Dr. McDonald says it's usually teens to 20s when you show signs. So hopefully that answers without Dr. McDonald being here.
Thank you.
Absolutely. So one question that arises is, why isn't our DNA the final instruction manual for these proteins? Why am I telling you that RNA is the final instruction manual? So to answer that question, I have given you a long stretch of an instruction manual that would be akin to your DNA. And you can see here that this doesn't make sense. It's not English. However, I am going to tell you that it is English and there is instructions in there. All we have to do is remove certain sections of this manual and suddenly the manual makes sense. If we remove 3 sections, the manual now states, step 1, screw bucket into arm. This process of making instructions from your DNA is called splicing. So your DNA looks like this, and your DNA makes RNA that looks like this. In order for RNA to work, it has to have parts of it removed for the instructions to make sense. The removal of parts of your RNA is a term called splicing. The variant that we have found in this family has this name. So this variant has been found to affect splicing, leading to an incorrect protein. So the process of making the instruction manual is incorrect. Notably, this exact variant has been described in another family that lives in Brazil. Interestingly, in this article, the family states that there is no family history of Croiteremia. We have since questioned whether this is actually true or whether there's individuals in the family similar to our unaffected brother that actually have this mutation and have eyesight, so don't realize that they too carry this mutation or this variant. So with this variant, I told you that it affects splicing. What happens to our instruction manual if we don't splice correctly? So in my example here, this is my DNA, which has now been made into RNA. And instead of removing the 3 pieces that I removed before, I have changed the last part, and I am now removing the word arm. And my instruction manual now tells me, step 1, screw bucket into roof. So you can see my nice digger here is definitely not going to work. I have made a protein, and I have definitely made a machine here based on these instruction manuals, but it's not the right protein. It is not the right machine. It is not going to perform the same function that it would have performed had we spliced correctly. So let's talk about the names of the players in this game. When we refer to the gene that is involved with Croiteremia, it's called CHM. Now, normally, when we talk about the protein that is produced, it has the same name as the gene. It does not for Croiteremia. It's actually called Rep1, the protein. And to be very honest, it actually kind of helps, because normally in biology, when we refer to the gene, we italicize the name. And then when we refer to the protein with the same name, it's just not italicized. So it actually helps a little bit in Croiteremia to understand what I'm talking about, because when I'm referring to the gene that can carry mutations, it is called CHM. When I refer to the protein or the active machinery that does the job, it is called Rep1. The clinical disorder is called CHM, and it has no italics. So that's the difference between when I'm referring to the disorder versus the gene. Okay, the jobs of the other players. Rep1, the protein that it's affected, has the important job of adding a flag to another protein called Rab27A. And I'm going to define this as the cellular equivalency of hanging a sign on a building. By adding the right sign, we can direct traffic to the right location. And so in this image, Rab27A is the building. There is the sign that's hung so that people know which building to go to. And here I have a woman named Rep1, whose job it is to hang the sign. In Croiteremia, the sign is a complicated molecule called Retinal. And this is a process called prenylation. And simply what it does is it allows Rab27A to go to the correct location in the cell. So Rep1 is like the woman with the hammer hanging the sign. Rab27A is the building. And we need the sign in order for Rab27A to know where to go. So step 1 of our interesting brothers. We realized quite quickly that we needed the unaffected brothers' samples. The cells would be preferable because these can be grown in culture. And from those, we can get both DNA, RNA, and protein, all of those 3 parts of the central dogma. Otherwise, we have to keep bugging this man for samples. And part of the problem is that we're in Canada, and this individual lives in Spain. And I told you that RNA is very labile. And by the time you get a sample from Spain to Canada, generally the RNA is not in very good condition. There may not be any at all. So we decided in our grant application to fund him coming to North America. Fortunately, Dr. Sang in New York offered to examine him and to take both skin and blood cells so that we could grow them in culture. It was actually at the beginning of the pandemic when everything shut down, and it was amazingly difficult to get an individual from Spain to North America. We actually had to write letters of support to the embassy. So this took many months in order to get him over. So we now have samples from the unaffected brother. We're still working on getting the cell lines from Dr. Sang, which will happen, but we are currently have samples that Dr. Sang sent to us so we can analyze them. So our very first hypothesis when we were looking at this, when we approach experiments, we always want, when we're spending especially hard-earned money that you guys have given us, we want to be as careful as possible and start with the inexpensive, obvious experiments before we get to the more expensive experiments. Obviously, if we got our answer right at the beginning, we could save as much money as possible. So our very first hypothesis was that the unaffected brother retained normal splicing, that for some reason, his bucket was still attaching to the arm rather than to the roof of his tractor. So we did look at that and found that indeed he actually does have abnormal splicing because we looked at his RNA, so that particularly labile. In a normal male, he has a dosage of 1, so he has a full complement of RNA. When we compared both of our brothers, we can see that they have very minimal full-length RNA for the Croiteremia, for CHM. And when we examined the RNA in smaller pieces, so this here is the full-length RNA, so the entire gene. When we just looked at a smaller piece of the RNA, we see that his RNA is indeed smaller. So this is called a gel electrophoresis. And in this, the RNA runs through, it's kind of just like a sponge. And the larger RNA takes longer to go through the sponge than the smaller RNA. So for our normal male, we can see that he shows up right here, whereas our affected and unaffected brother are together and run through this gel quicker, meaning that their RNA is smaller, which means that for sure the instruction manual is not correct. Okay, so hypothesis number 2. Possibly the unaffected brother has the CHM protein, reminder that that's called Rep1, that is more stable. So we know that he doesn't have RNA or very much, but perhaps the protein that he's making sticks around longer than it does in the unaffected or the affected brother for some reason. So the unaffected brother possibly produces protein that sticks around, whereas the affected brother has protein but is degraded right away. It's transient. And the result from that was that no, the protein is not present. It is the same as in his affected brother. So we can see here this arrow of Rep1, the protein. In a normal male, this is where the protein would show up. We can see here for our 2 brothers, that neither of them have Rep1. This is just a control protein to show that we ran the gel correctly and that our experiment worked. Like I said, we got cells from different parts of the body. This is a lymphoblast, so this is your blood. So he does not have the protein in his blood as a normal male would. And he also does not have the Rep1 protein in his skin when we obtained some protein from his skin. So much for hypothesis 2. Neither the affected or the unaffected brother have the Rep1 protein. Moving to hypothesis 3. In your body, there are two proteins with similar function. They are called, not surprisingly, Rep1 and Rep2. Rep1 is known to be present in the retina and function highly in the retina. Rep2 is known to not function in the retina. And this is why Rep2 cannot replace Rep1 for individuals that have Croiteremia. So we thought perhaps Rep2 is produced in very high levels in the unaffected brother, so that perhaps if we overproduced the second protein, it would make up for the lack of Rep1 and fill in. The result from this experiment was that no, Rep2 is present in the same amount in both brothers. Of course, this does not rule out that Rep2 is functioning differently, it's slightly different or present in higher amounts in the retina, because clearly the unaffected brother would not be very happy with us sampling his retina. So we haven't been able to examine that. All we can do is look at his skin and his blood. So from here, we can see that the Rep2 protein levels are not significantly different. In fact, if anything, it wasn't significant when we did the statistics, but the affected brother actually has less Rep2 protein. So much for our hypothesis that he might have more. Hypothesis 3, or maybe this is actually supposed to be 4, I apologize. Does the unaffected brother still retain the activity that Rep1 does? So this is the complicated process that I mentioned before called prenylation, which is analogous to the hanging of the sign. So it doesn't mean that Rep1 would actually be doing the activity, because we see that Rep1 is gone, but it would tell us that the activity is still happening. The sign is still there. Somebody else is hanging the sign. So this week, Alina is working on looking at the activity levels in the affected versus the unaffected brother to see if the unaffected brother still retains the activity, which is what we are now hypothesizing. Assuming that our unaffected brother retains the activity, we need to find out why. This is going to lead us to the more expensive experiments, whereby we are going to sequence both brothers, their entire genome, and hopefully if we have enough money, their RNA genomes to look at the differences. The DNA genome will allow us to look and see if they've inherited any differences. When we look at the RNA, it tells us whether the changes in the DNA have also resulted in changes in the RNA. So it allows us to see step 2 of that central dogma. So that's a little bit more powerful than just looking at the DNA, but making sure that the DNA properly made the RNA, and is there any differences? So the building that Rep1 works on, which I mentioned to you before was called Rab27A, is not really liked by the other similar protein called Rep2. So Rab27A is known to be very active in the retinal pigment epithelial cells that are responsible for transducing light. And we know that Rep2 doesn't really bind well to Rab27A. However, what if our unaffected brother has changes in his Rep2 or Rab27A that make them now suitable partners? So in my hypothesis, we have Rep2 as it normally exists. And you can see here that they don't bind well. They don't really like each other. And so because Rep2 doesn't fit well with its partner, we're not able to prenylate or hang the flag. However, if for some reason the unaffected brother has a slightly different either Rep2 or Rab27A that causes a slightly different shape, it means that they would fit together nicely and that RAB27A could undergo prenylation or the hanging of the flag and be done by REP2. So in essence, you wouldn't need REP1 because REP2 is now able to perform the function that before only REP1 could perform. Until now, it is thought that REP2 could never, ever replace REP1. But it is an interesting thought that perhaps REP2 is able to replace REP1. So our long-term hopes are that this unaffected man is clearly biologically cured himself. And if and when we determine how this is happening, it is going to give us the strong potential to make this happen in others. The part, even though we've had negative results and haven't found the reason why they're different thus far, I'm actually happy about it, which sounds a little bit surprising, but it means that because both of these men carry the same variant, both of these men just look like traditional CHMers in that they are missing their REP1 protein. It means that biologically right now, the male that can still see does not look any different than males that can't see. And this is a good thing because it means that there's something else replacing that function in his body. So it suggests that there's another pathway or another way to compensate for sight that we're previously unaware of. His mutation isn't special. His mutation is not causing REP1 to function or to even be present. If that was the case, then it would be much more difficult to predicate a cure or a treatment because he would truly just be a different type of CHM. But right now, every assay we have run, he looks like a traditional CHM male, and yet he is still retaining his vision. So we are hopeful that when we identify what that second pathway is, that we can then take advantage of that second pathway that science has never yet discovered and allow that to be used as a therapy for Croiteremia. So this would make the solution for Croiteremia possibly REP1 independent. So it wouldn't rely on the REP1 protein, but biologically on another protein that's actually working in the body. So I hope that explains what we're doing. We've had numerous stumbling blocks through COVID, but we are back up and working. Our institution has been shut down numerous times, as I'm sure, and we've had supply chain issues like the rest of the world. But we are geared up and working and about halfway through this grant. And so we really want to thank the Croiteremia Research Foundations in both the Canada and the US for funding this work. We want to thank Dr. Sang and his lab for seeing that unaffected brother in New York during COVID and for taking his cell lines and growing them for us. And I want to thank Elena, who's on the call, who's my dear tech, who's doing all of this, and Dr. Ian McDonald, who's my partner in crime, or actually my partner in research. And I'm available to take any questions about this, or we can chat about RNA and COVID vaccines. But if anyone has any questions about the work that I'm doing on Croiteremia, I'd love to have questions.
Let's stick with Croiteremia. I think we're also tired of the other one, right?
Yeah.
Here, we do have some questions. So now's the time, folks. Start entering your questions. I'll start sharing them with Dr. Hume. So can you tell us more about the next steps? What or if the hypothesis, if this is true, what are the next steps?
Yeah.
And then if it turns out to not be, are there other concepts in the wings that you're going to try?
Yeah, great question. So we're currently going to send off the samples from both brothers to another lab that does, we'd hope to have a local source for doing whole genome and whole RNA sequencing, but it looks like we're going to have to send that off, which is fine, to a lab to do that, an external lab, and pay for that service. So when we get that back, it's a lot of analysis. It's a lot of genes and a lot of manuals to go through to see where their instruction manuals are different. So we have some ideas of where we want to look first. We obviously want to look at the pathway that we know, that REP27A, RAB27A pathway, to see if they have any differences there. If we do spot differences there, which we likely will, then we have to test that in a cell culture. So we have to see if we can generate the same mutation in a cell culture and look at our assays, our prenylation assays, to see that our prenylation went from nothing to being fully prenylated. So when we look at those in vitro assays, after we see the differences in the whole genome and whole RNA sequencing, then we'll be able to mimic what we think is happening in the body in the test tube. Once we've confirmed it in the test tube, we now have a pathway that we are going to target in the body. So hopefully that makes sense.
Would that have to be done in the animal model first, or could you go straight to human?
Great question. We always prefer to do it in the animal model first. However, if we find that there are known safe drugs that target the pathway already, it is conceivable that it would go straight to humans. If we are talking about identifying a brand new drug or new treatment to upregulate this pathway, we'd have to first do it on animals. So it really depends on what we find.
Gotcha. All right, here's another question. It says, hi, thanks very much for your research. I have a question. Would you consider helpful to generate the unaffected brother's retina from his stem cells in the lab and then test his REP1 protein production from this retina cell?
We could do that. That's an even more expensive experiment. And so it's definitely not off the books, but what is cheaper right now is to look at the differences that we are seeing specifically in the RNA, because it will allow us to see which pathways are different. And if we have a hint that it actually is, for some reason, even though all of our experiments are not suggesting it's REP1, if we are seeing a difference in REP1, we could go back and do that. But right now we'll look for obvious differences between the brother that's affected and the unaffected brother to see which pathways are different.
If you're seeing that your hypothesis, the fourth one that you're describing now, would come to fruition, is that something that would be like an eye drop topical? Would it be injection? Would it be orally taken? What, if you were to play this out, how does it look?
Anytime that you're doing any treatment, the difficulty is the permeability and targeting to the retina, obviously. So, you know, I'm going to say that an eye drop is probably a better way to go. Interestingly, I'm now working with another group that's doing nanoparticles and doing drug encapsulations. So I'm hoping at the end to tie the two together. This other work that I'm doing has shown that these nanoparticles actually are very biologically safe and are absorbed into the cells. So I'm hoping to actually, in the end of this research, is to tie the two together and use my nanoparticle work to be able to encapsulate a drug. And it's different than the gene targeting that used a viral vector, because that was virus. This is simply a synthetic capsule that is very tiny and is safely taken up by your cells. So what I'm hoping to do is be able to encapsulate a drug, put it in drops, and you drop them into your eye. Alternatively, if it wound up needing to be a intraocular injection, that it would be a one-time ocular injection, and the drug would be quickly taken up by your retina and able to preserve what RPE, retinal pigment epithelial cells, that you have left.
Wow, wow. Okay, here's another question.
Yeah.
The patients you have, one brother is totally unaffected. What about those that are affected, but it is mild? Have you got any reasons for why this may happen? I have two sons, one 4, who is not showing any signs of CHM, whereas my 8-year-old is already showing pigmentation and night blindness.
So the exact reason why people show changes at different ages is not yet known. We don't know why some people, and just like we don't know why some female carriers are worse than others. And truthfully, the fact that this brother is unaffected could suggest that there is another pathway that could be playing a role in how soon people are affected. Maybe some people have this pathway and don't realize it. And for example, that Brazilian family that I showed in that first article, where they say they have no known family history, as we all know, CHM is X-linked, and it would be very unusual unless he's adopted to have no family history anywhere along the line. So perhaps there's another pathway that, of course, if you have vision, you don't go in for, you're not even eligible for CHM gene testing. So it's not like we've widely gone around and tested a lot of people with perfect vision to see if they have any CHM gene mutations and have evidence of a second pathway. So we're still hoping that there is a second pathway that will explain both this unaffected brother, as well as the different degrees of clinical presentations that can exist in families and in female carriers.
Can you give us an idea of research costs?
Yeah, research costs. So probably the biggest costs are hiring your technologists to the job. And let me tell you, they go through school for many, many years and are really not paid what they're worth. I think a lot of people in society aren't paid what they're worth, but for sure, research techs are not. The costs have gone up unfortunately, with the pandemic, because in research, we use the same plastics and the same materials that anyone does for doing COVID detection in the pandemic. And so it's supply and demand, right? Costs have gone up. So, and sometimes you can't even get the equipment to do the costs. For doing the whole genome and whole RNA sequencing, we're probably looking at a cost of, $3,000 to $4,000 just for another lab to take that, because it's a very expensive process. And then we would get that back and do all of, and we're doing it on 2 brothers. We're not just doing it on 1 brother, we're doing it on 2 because we need to see the differences. So yeah, minimally, I say $3,000 to $4,000 just for that single experiment.
We have a gentleman, Eric, here, who is joining us from France. Hello. He's asking, it's more of an internal CRF question. Is it possible to have a written synthesis in English and then translate it into French? So I just said that, and I'm sure Kathy, our executive director, and our team will get on that. We probably have an answer for you maybe by the end of this video. Otherwise, we'll get back to you for sure, Eric. All right. I know you don't have a magic ball. We always try to, we always precede that, at our conferences. We always say that to our researchers. We know you don't have a magic ball, but But given where the research is currently, do you think there will be a treatment in the near future? Are you looking 5 to 10 years? What's your timeline?
You know what? It is such an exciting time in genetics, and given the advances that we are making. So I can tell you about another disorder called spinal muscular atrophy, for which now, and it is also a splice disorder, and that's why I'm pulling that one out. It wasn't a random pull, for which there now is a treatment for it. So I do think that it is on the horizon. I do think that there's much greater awareness, and funding agencies are really now devoted to funding translational research. So just like this, where we have found something in a patient and now we're trying to bring it right to the bedside and be able to help patients. So I think it's, yes, to be very honest, I do think we are going to see in the next 5 to 10 years, some big changes come about for sure.
Are you seeing that it would stop the progression, or do you think it would have the capability of returning lost vision?
Good question. I think at first we are going to stop the progression, because I think that's the easier thing to do, is preserve the retinal pigment epithelial function. So keep that prenylation happening. To reverse it and actually grow retinal pigment epithelial where it has already been depleted is going to be much harder. So I think stage 1 will be arresting disease progression for sure.
Brandon's asking the question, do you know if the mother of these CHM brothers is displaying any unique traits as compared to other female carriers? Good question.
It's a great question. And I've asked Dr. McDonald, and Alina, I think, is on the line and can pipe up, but I think I recall him saying, no, she's presenting exactly. She has signs of being a carrier, just like any other female CHM. So she doesn't look unique. Yeah, there's Alina.
Yeah, Alina said that, correct. Typical carrier signs.
My memory's still good.
Okay, let's give it one more minute. If anybody else has any final questions or thoughts, here they come. In your research, will that be your points of reference to stop the progression first?
Yes, it will be. It also will be. What I think we could do is obviously stop progression, but if you knew that you had a family history and we could treat babies at birth, or even prior to birth, I think that would be the best. So, I can envision a woman having prenatal testing done. She finds out that she's carrying a male, 50% chance of having the bad X chromosome, the one with the mutation. And then we start either treating that baby right from birth so that the prenylation is not affected at all, and that child doesn't even actually ever know that they would have lost retinal pigment epithelial activity.
It's a game changer, right? Potentially.
I mean, this for us, like for me, this finding of 2 brothers that are very discordant is incredible, because biology has really given us the answer. Biology has told us there is a fix for this, and we just need to figure out what it is.
Is he taking really good care of himself? Do we need to support him to make sure?
Well,
it's any dangerous activities.
It's a great question, and even we've even contemplated things in the lab, like, is he eating something different? Is his diet different? Is his environment different? I mean, of course, it can be anything like that. Seeing that he has no Rep1, for me, implies that it's not diet. He's not done anything different than the affected brother, because he looks the same. If we mix those samples up right now and I ran them out, I wouldn't know the difference. They look identical. So that part has me more excited, because then he looks like any other person that has Croiteremia. He's not this really unique, it's not that only this mutation is going to benefit from this, and that's some of the cures out there, is you need to have a particular mutation in order to get the cure. Given that he has no Rep1 and almost all of the CHMers have no rep1, he looks identical.
Hmm.
So I'm really excited that it could be a cure for everyone and not just for somebody that has this exact mutation.
We have one of our fine doctors on the line with us too, Dr. Richard Lewis, and he commented application prenatally has been done for X-linked ectodermal dysplasia in utero.
Wow, thank you. I didn't know that.
Let me get a big thank you shout out. And then would you consider treatment for children at trial stages?
And it depends on, for sure, but it depends on what we find. If we can find a drug that is already a known target of that pathway, and the drug has already gone through the phase 1, 2, and 3 clinical trials, phase 1 and 2 showing safety, then absolutely.
Yeah.
Because we know it's safe. I think, ethically, we can't, Croiteremia isn't a life terminal illness. And so we definitely wouldn't want to start, give somebody something that would end their life or has safety concerns. A little bit different than when you're treating like an end stage cancer patient and you can take a few more risks. We wouldn't want to do that with somebody that has a full life to lead ahead of them.
Let's see. We have two more things quickly for our friend in France, Eric. The person you could try using free Microsoft Translator, which is an app, which might allow you to play the recorded video link, but we'll also be sending it out, and try it that way. And if you're still having problems, you can let us know for sure. Let's see. All right. Thank you for your time. I appreciate that. We have a couple more questions. Is there a way that we can get updates on this research as it progresses? Absolutely.
Yeah, I'm beyond excited about this project. So I absolutely want to share with you guys what's happening. I mean, I'm hoping you guys are as excited as I am about this.
I think we are. Absolutely. I'll speak for us all. Are you going to be able to join us? And pardon me if you already told me, are you able to join us in June at our conference?
I just wasn't sure with COVID what was happening. So yeah. If it's virtual, absolutely.
Well, it's going to be actually in person.
Okay.
So we'll talk more about that. I'm sure we'll talk more. Let's see. Here's another one from Vince. I am one of 7 sons and the only one with vision loss. How can my brothers get gene testing to know if they have the CHM?
Yeah, that is a good question, because up until this point, we would have said that if they haven't shown signs by this age, that they don't have the CHM mutation that you have. Given that this has happened, there could be other people walking around that carry the mutation that are not showing signs. It's difficult because I'm not sure with funding, if funding will allow the testing to happen without showing signs or symptoms. So in essence, it's kind of a backwards world, isn't it? Because we don't look unless we have a reason to look, but because we don't have a reason to look, we don't know. And so we're kind of caught in this vicious cycle of there's no funding to test unaffected people, which unaffected people are actually sometimes, even more interesting than affected people.
But they should be able to get a genetic testing done and there's information on our website about how to do that. So I would tell Vince to go to the website and take a look, because you certainly should have everybody tested.
Yeah, it just might have to be self-pay. The insurance company may not pay for it without them having traditional signs and symptoms of CHM. But for sure, I'm sure you can get gene testing if you're willing to pay.
Dr. Lewis had one more question here. Has his mother, maybe he means their mother, but has she been examined?
I believe so, Alina. The mom has been examined, right?
And then how did you find these 2 brothers? Yes, we have images from the mother.
The brother saw Dr. Sang in New York, and I'm not sure why the unaffected brother came for testing. And maybe Alina may know the background of why the unaffected brother came for testing, because that is a good question that I've never thought of, is why we received samples on the unaffected brother.
I'm going to go back a little bit. Dr. Lewis was saying the man who he's one of 6 or one of 7. And then real quickly, there is a link in the chat. I think it's Vince, right? There is right here, Kathy Wagner posted, it is myretinatracker.com. But that would be something that you should, it's a link right here in the notes you can click on.
Yeah, Alina, it's coming back to me. Yes. So this variant, prior to having it, we are now classifying this variant as pathogenic, but prior to this, we actually classified this variant as of unknown significance. That means that we didn't know if it was the cause of CHM or not. When you find a change, we all carry changes in our DNA. I have green eyes, you have blue eyes. There's changes in our DNA that cause that, and it doesn't mean that we have a clinical condition. So we don't know whether a change that we see in the CHM is just a normal everyday change that people carry, or if it's something that's pathogenic. Often we have to look at the RNA, and by looking at the RNA, we can see that for sure the instruction manual is not right. Looking at the RNA is difficult. It's not done every day in diagnostic labs because RNA is labile and difficult to work with. And so because this variant initially was unclassified, we didn't know what it meant, we went on to look at the RNA. So we did collect samples from other family members to examine whether this variant was pathogenic or not. And that's how this kind of incidental finding came about, that we found it in the unaffected brother.
So Dana, I want to answer your question. She's asking if this recording will be made available, and it certainly will. We will put it on our YouTube channel. We'll be sending out a copy of this. You can find it on our website. There's going to be multiple ways. It'll take about a day or 2 to get it off the cloud, but we'll be able to share that. Is there a link to your research?
No, I don't have a link to my research. Alina, I'm not sure if Ian has a link that we could put, but generally the best way to keep up on the research is, well, either A, that we give you updates and talks, or B, we do publish the research when we're at a point that we're able to put it into the literature. So we're not there yet for this project, just because we still don't know the solution yet. But the minute that we have a hint as to what the solution is, you should be able to look up my last name and Ian McDonald and Alina on here, and you'll see that we've published it. But for sure, you guys will know that.
And we will too.
Yeah, you guys have
sorry. Yeah, the foundation absolutely will be giving updates as we do in our newsletter and on our website. Now, you started the funding, correct me if I'm wrong, but in 2020, right? And was it a 2-year or 3-year proposal? I should know.
I feel like it was 1 year at the time, and then we had to get an extension because of the COVID. And I can absolutely tell you that we're halfway through the funding right now, like spending. We've got about half of it left in the account. So it'll be used up by June, by the time the deadline came up. Like this work that I'm telling you right now will be done by June.
And I think it's really important for our audience to understand too, when our science advisory board reviews these different grants, we have a great science advisory board who reviews all of these. And when we agree and the board funds research, then in the agreement with our researchers, you need to provide updates. And so we as a foundation, we have a science advisory board that gets these updates, and they ask the pertinent questions back to the researcher if we have our board members have specific questions. So the foundation, you can rest assured that the foundation and our team of very professional doctors and researchers are asking those questions and keeping a pulse on it.
Absolutely. And I have to say, researchers are like we're pretty stingy with our money because it's really hard to get. So we really take each dollar and use it very carefully when designing our experiments. As you can see, we've done the really cheaper experiments first. You don't ever want to go and blow $4,000 is a lot of money, and you don't want to blow that right at the beginning of your experiment. You need to really make sure that you're doing that in a logical progression. And I see another question of how much more funding would we need to continue. So what I'm hoping is that when we get the answer back from the whole genome and whole RNA sequencing, that we can then design experiments to look at what we're seeing and what the differences are. And then we could put in maybe another application with a budget of, okay, this is what we found. We have found that Rep2 might be working. We want to now try to upregulate Rep2 in test tubes just to see if it works. So I think we'll have a better idea shortly in the next upcoming months what the next steps are, and then what sort of funds we would need to fund that. But
that's one of the great things, that a lot of our members are great fundraisers, and they do family fundraisers. They join us in the $1 Million Bike Ride, which is through UPenn.
Yep.
There's just a variety of ways people raise money, or they make a monthly donation, or they do all sorts of things. So funding has not typically been a problem. But we're very judicious in how we choose. And again, our science advisory board helps us make those decisions.
Yeah.
But
and I want that people to be rest assured that when the money is given, that researchers are notoriously very, very careful with how they spend that money, because we really appreciate it and try to stretch it as far as we can and make sure that every little thing that we're doing counts. So we know that this was a lot of work to raise this money, and definitely want to put every dime to good use.
Well, this has been really encouraging, this time spent with you today. It's been kind of a hard season for us. Mm-hmm. Oh, hey, Martha Hillier, hello from Canada. Would this be considered a stop codon variation? She comes in with one of those science ones right at the tail end.
Okay, I can do it. It isn't your traditional stop codon, but the answer is that yes, it does cause a stop because the protein is not fully produced. So the instruction manual is terminated prematurely because of the splicing, not because the change introduced a stop. So I think biologically it has the same effect at the end, but the mutation isn't exactly the same. So that's, I think, the part that I was excited about, is that given that the mutation isn't the same, but we are seeing the same biological effect in that a traditional stop codon makes Rep1 absent, and this splice mutation does eventually introduce a premature stop codon, and it does make Rep1 absent. So we're seeing the same biological function. So it's not going to be a cure based on a mutation. So that is the best news, that really, when you look at cystic fibrosis, you have to have certain mutations in order to be on the drugs. Spinal muscular atrophy is the same way. And so ideally, it's really great if we can find a cure that's not dependent on the mutation that you have.
This kind of gets back to the foundation that some of our researchers are trying to get at. With gene therapy, we've tried some other things that are advanced, if that's an appropriate word.
Yeah.
This is kind of getting back more towards the disease process itself, the foundational aspects. And so this is super exciting in a season that's been hard for the CHM family and community. It's been rough. Yeah. So with the studies that have come out, so this is extremely encouraging, and we're just so super grateful to everything that you and your team are doing. Of course, you know how much we love Ian McDonald and how he's always going to be the godfather of our organization, if I can say that.
I adore him too.
Yeah, he's great.
Yeah.
Well, thank you so much. And we'll keep everybody updated. We will be sharing this video, and thank you again, Dr. Hume, for everything that you're doing. And we're looking forward to maybe seeing you in June.
Okay. Thank you guys. I really appreciate it. And we will definitely keep you updated.
All right. Thank you so much, everyone.
Thanks,
have a good rest of your week. Stay warm.
Yeah. Thank you. Bye.
Okay. Goodbye.